Ayun, recording na. So, good afternoon ulit. Naririnig niyo ba ako?
Yes, ma'am.
Okay, thank you. Nakita niyo ba yung slides ko?
Yes,
Opo.
ma'am.
Okay, thank you. So, let's start na. Our lecture for this afternoon is going to be about DNA repair and recombination. So, last time we discussed about DNA structure and DNA replication. So, ito pala yung, bago tayo magsimula, ito yung kung bakit ko kayo ini-encourage na mag-aral at your own pace kasi we all go through things differently. And if you ask one, how deep is the mud, iba rin yung na-experience nila kasi yung iba sa atin, different rin yung resources and meron tayong different challenges rin na um, na-encounter habang nag-aaral tayo in this pandemic, right? So, kaya ko kayo in-encourage na lang na study at your own pace and answer your quizzes at your own pace. And also, yung requirement ko sa laboratory, wag nyo naisipin yung deadline nun. Lahat ng bibigay ko ay ang deadline for last week ng SEM na to. So, ayun. Pero mas maganda kung on time nyo pa rin ipasa para hindi maipon yung gagawin nyo. And para hindi kayo mag-cram sa pagsagot ng mga quizzes or nung forms na binibigay ko or nung activities na binibigay ko. So, ayun. So, yun lang bago tayo mag-start. Kaya tayo in-encourage ko na study at your own pace. Ayun. So far, we have studied the hereditary information in the form of the DNA, right? And the structure. So, alam na natin yung structure ng DNA and how DNA replicates. And now, we are going to proceed to answer how DNA is repaired when it is damaged. So, lecture 3 is about DNA repair and recombination. Okay? So, our objectives is to learn the DNA, to learn how the DNA is damaged and repaired, to learn the various DNA recombination mechanisms, and to study diseases related to dysfunctional DNA repair systems. And TikTok to actually. So kung kilala niyo si Darion Nguyen sa Lab Shenanigans, magaling siya gumawa ng TikTok about scientific stuff. So di nagpe-play yung video pero basically nag-TikTok siya tungkol sa eukaryotic DNA polymerase daw na gumagawa ng errors every 10,000 to 100,000 nucleotides. So tingnan niyo yung TikTok account niya or yung Twitter account niya. Marami siyang videos tungkol sa laboratory stuff actually. And we have learned that DNA polymerase has a proofreading mechanism, right? That is why the mutation rates for both bacteria and eukaryotes is low, diba? So if you remember in our previous lecture, the mutation rates for bacteria and eukaryotes is low because the DNA polymerase has a proofreading mechanism. And to simplify, the proofreading mechanism in DNA polymerase. So when the DNA polymerase adds an incorrect nucleotide to the new strand of the DNA, it detects the bases which are mispaired. So itself, meron siyang capability na ma-detect niya yung pagkakamali niya. And then, it performs an exonuclease activity, removing the wrong nucleotide. So, yun yung DNA polymerase proofreading mechanism, right? And aside from the proofreading mechanism of the DNA polymerase, there are various ways which the DNA is actually repaired. So, in this lecture, we will look at each of those repair mechanisms. But first, we will take a look at how DNA is damaged. So, itong itong figure na to, actually, experiment yan ng medical or researchers from Harvard School of Medicine. So what they wanted to observe is that how the DNA repairs itself. So makikita ulit natin yung figure na yan mamaya. In a certain repair mechanism called non-homologous end joining. Pero mamaya, titingnan natin. So there are various ways how the DNA is damaged. First is through oxidative damage and hydrolytic attack and methylation. So, ito lang naman yung um, portions of the DNA or the nucleotides which um, gets damaged. So, yung blue arrows, wait, yung red arrows muna pala, nagkakaroon doon ng oxidative damage. Yung blue arrows, nagkakaroon ng hydrolytic attack, and yung green arrows, pinapakita lang yung kung saan nagme-methylate. So, for example, here in guanine, in a certain part of the guanine, so yung oxygen atom dito, name-methylate siya due to harmful chemicals. So, when harmful chemicals react to the DNA, minsan nagkakaroon ng methylation. So, ito yung example. For guanine, nagkakaroon dito ng methylation, and that methylation is considered as damage. Nadadamage yung DNA mo. And another type of DNA damage is called the depurination. So in depurination, the glycosyl linkage of purine to deoxyribose hydrolyze. Sabihin nyo kung mabilis yung pag-discuss ko, okay? So, ito. So, sa depurination daw, the glycosyl linkage of the purine to the oxyribose hydrolyze. So, how? So, for example, we have here a guanine or a nucleotide with guanine as its base, right? Na-remember nyo ba yung structure ng nucleotide? So, ganito yun, di ba? So, phosphate group, sugar, and your base. So, sabi daw dito, in depurination, the glycosyl linkage of purine to the oxyribose hydrolyze. So, your purines are, again, ano yung purines nyo?
Adenine and guanine.
Yan, correct. So, correct. Your purines are adenine and guanine. So, for example, here you have your guanine and na-hydrolyze daw yung guanine mo. So, when your guanine is hydrolyzed, it displaces the guanine base. So, now, yung nakakabit dito na guanine, nawawala. And magkakaroon ka dito ng hydroxyl group. So, yun yung depurination. Basically, through hydrolyzation, nawawala yung purines mo, the bases. So, yun yung depurination. So, remember again, your purines are your adenine and guanine. And here, as you can see, depurination, it leads to mutation. So, for example, you have a depurinated adenine here. So, ibig sabihin na wala yung adenine, right? And as you can see here, when the DNA replication proceeds, one of the daughter DNA nagkakaroon ng deletion. Nadidelete yung base pair dito. Di ba dati dito merong adenine and thymine, but then the adenine is depurinated. Therefore, yung daughter DNA niya nag-mutate to have a deletion mutation. So, na-delete dito yung base pair with adenine and thymine. Okay? So, yun yung mutation na nakakos ng depurination. Another type of DNA damage is deamination, which is the conversion of nucleotide to a natural product by hydrolysis of the amino group. So, dito naman, yung na-hydrolyze ay yung amino group in your nucleotide. So, for example here, your cytosine naman, or your nucleotide with a cytosine base, ito yung amino group, right? Ito, NHH, amino group yan. And na-hydrolyze daw yung amino group. So, kapag na-hydrolyze yung amino group, magbabago yung chemical composition ng base mo. So, ito, from cytosine, naging uracil, yung base mo. So, yun yung deamination. Na-hydrolyze mo yung amino group in your nucleotide. So, here are the specific deamination in each of your nucleotides. So, here is your purine. So, adenine and guanine, right? So, here in adenine, ito yung amino group niya and na-hydrolyze. Therefore, mababago yung itsura niya, magiging siyang hypoxanthine. And this is considered as unnatural product, unnatural, UN, a natural product. Hindi siya natural kasi um, hindi siya yung natural na nag-o-occur sa DNA strands mo. So, again, another, as you can see here in your guanine, meron kang amino group, right? And once this is hydrolyzed, magkakaroon ka ng a natural product called the xanthine. Ayun. So, yan yung deamination na nangyayari sa purines mo. Okay. So, ito naman yung deamination na nangyayari sa pyrimidines mo. So, your cytosine and thymine. So, in cytosine, again, this is your amino group. And once it is hydrolyzed, yung cytosine mo, mako-convert into uracil. Okay. And again, this is considered as a natural product kasi hindi naman nag-o-occur ang uracil in your DNA strand. And if we take a look at thymine naman, wala raw deamination. Bakit kaya walang deamination sa thymine? Alam niyo ba kung bakit? Tingnan niyo yung figure. Bakit walang deamination sa thymine? Mhm. May nakakaalam ba? Hmm. Okay. So, sirit na ba? So, yung thymine kasi, kaya siya walang deamination kasi wala siyang amino group. So, balikan natin ah yung definition ng deamination. Conversion of nucleotide to a natural product by the hydrolysis of the amino group. And if you look at your thymine here, wala namang amino group sa thymine. So, therefore, hindi siya nade-deaminate. Okay? So, walang deamination sa thymine kasi wala siyang amino group. So, dito, merong amino group. But your thymine, it does not have an amino group. So, walang deamination. Okay? So, the amination is a DNA damage which causes mutation in the form of substitution. So, as you can see here, ito yung DNA strand mo. Once your cytosine is deaminated to uracil, na-deaminate yung cytosine mo, tapos nagbago yung structure niya, naging uracil, yung daughter DNA mo will have a mutation which is called substitution mutation. Kasi, imbis na cytosine yung nakabind dito, or imbis na adenine yung nakabind dito sa daughter strand mo, wait, sorry. Imbes na guanine yung nakabind dito sa daughter strand mo, adenine. So, 'di ba ito yung template strand mo. So, yung template strand mo, tapos merong mutation, uracil. So, yung new strand mo ngayon, imbes na guanine yung malalagay, adenine na. Kasi uracil yung nakalagay doon sa template strand mo. So, that mutation is called a substitution mutation. Okay? So, dahil sa damage na cause ng deamination, meron ka ngayong mutation which is called your substitution. Okay. So, another type of DNA damage is called your pyrimidine dimers. So, pyrimidines, again, are CUT, right? I cut the pyramid. So, cytosine, uracil, and thymine, they can form pyrimidine dimers. And in pyrimidine dimers, it happens when your um, nucleotide bases, they act or they are exposed to UV light. And yung UV light mo, it causes covalent bonds between your pyrimidines. So here, as you can see, this is a thymine dimer because these two bases are thymine. And merong covalent bond sila. And this pyrimidine dimers, it causes a kink in your DNA strand. So, kapag meron kang pyrimidine dimers, meron ka bigla ditong na covalent bonds, tas masisira yung structure ng DNA mo. So, imbes na maayos yung connection ng DNA mo dito, nawala yung hydrogen bonds mo dito, nagkaroon ka ng covalent bonds between your pyrimidines. In this case, your thymine dimers. So, ayun. Yun yung pyrimidine dimers mo. Wait lang. Mhm. And another type of DNA damage, or like yung general term for the DNA damages that happens in your cells are your single strand breaks and your double strand breaks. So in merong general forms ng damages, which are your single strand breaks and your double strand breaks. And mostly they occur through um parang through exposure to X-rays or gamma rays. So as we can see, merong maraming types ng damage, right? So, yung nakita nyo kanina, merong oxidative damage, merong hydrolytic, merong methylation, merong deamination and depurination, and meron din formation of dimers. So, ngayon, when these damages are not repaired and they accumulate, diseases will occur. So, wala na, finish na. Kasi kapag nag-accumulate yung damages mo in your DNA, mahirap na yung DNA mo mag-function properly to transfer its hereditary information and to um, parang magkaroon ng maayos na process within your cells because again, your DNA is it contains your hereditary information and um yung genetic information mo, yung genes mo which produces the different kinds of proteins which are responsible for all your cell processes. So if these DNA damages are not repaired and they accumulate, diseases will occur. So ngayon, titingnan natin kung paano itong damages na 'to are repaired. Pero actually, the DNA double helix is readily repaired. So despite all of these damages, hindi sila nakakapag-accumulate, 'di ba? So remember nyo doon sa previous lecture natin, merong low mutation rate. Kasi nga dahil dito, dahil yung DNA mo readily repaired siya. So how? Your DNA has two strands, right? So if one of the strand is damaged, it can use the other strand as a template for repair. So as you can nakita nyo kanina, 'di ba, nung deamination, napalitan yung cytosine to uracil. But then pwede mo yun ma-repair by using your template strand. So, pwede mong ipalit dito, kunyari, yung tamang nucleotide base na mali. So, makikita natin yung types of repair which happens in your DNA. So, first is your translation polymerase. So, in our previous lecture, we've learned about the inherent proofreading mechanism of the DNA polymerase, right? So, merong isang type ng DNA polymerase called the translation polymerase, which it switches to the it switches with your DNA polymerase and replicates past the damage. So, the replicative polymerase is switched out for specialized DNA polymerases and replicates past the damage. And yung translation polymerase mo in eukaryotes is called your DNA polymerase ETA. So, so yung DNA polymerase ETA mo, ito yung translation polymerase mo sa eukaryotes. So, as you can see here in the figure, gumagawa ng new strand yung DNA polymerase mo, right? But then, when it encounters a damage here, yung DNA polymerase mo magsi-switch with your translation polymerase. And your translation polymerase, it still replicates past the damage by adding the right nucleotide base. So, ito rin yung tinatawag na, ano nga tawag doon? Damage tolerance. Kasi kahit na may damage dito, natotolerate pa rin ng translation polymerase and it replicates past the damage. So, for example, yung damage mo dito actually na nakikita niya ay yung thymine dimers mo. For example, meron kang pyrimidine dimers and specifically thymine dimers. So, yung thymine dimers mo ngayon, 'di ba thymine sila. So, yung tamang magbabind doon ay adenine, right? So, adenine binds to thymine. Now, your translation polymerase, it has an ability to add adenine nucleotide dito sa growing strand kahit na merong thymine dimers dito. So, yun yung translation polymerase mo. And dahil doon, meron ka ngayon na tinatawag na damage tolerance. Okay? So, ngayon, ano yung next? Wait lang. So, now let's take a look at the specific damages responsible to repair single-strand breaks and double-strand breaks. So, mag-start tayo with single-strand breaks. So, first ay yung mismatch repair. Second ay yung base excision repair. And the last is your nucleotide excision repair. So, let's take a look at each of these in a closer look. So, isa-isayin natin. So, your single-strand break DNA repair, for mismatch repair, um, it actually is an effect of genetic defects which, for example, yung Lynch syndrome mo and makikita i-di-discuss natin 'to mamaya. Wait lang, may nag-chat. Nawalan ng internet. Okay lang, ipo-post naman natin yung, um, lecture natin. So, ayan, nasaan ako? Mismatch repair. So, mismatch repair, it repairs parang related siya sa genetic defects such as the Lynch syndrome, or as you will see later on, this Lynch syndrome is your hereditary non-polyposis colon cancer. But we will discuss this later. So, yung next naman ay yung base excision repair mo. And the base excision repair is attributed to carcinogenic exposures such as tobacco smoking, kapag kumakain kayo ng red meat masyado, yung longganisa, yung hotdog, may mga carcinogens doon. Or parang kapag exposed kayo in pollution. So, ganun. So, yung base excision repair, attributed siya in carcinogenic exposures. And DNA damage caused by carcinogenic exposures. And lastly, your nucleotide excision repair is attributed to damages caused by the UV light, or yung nakita nyo kanina, yung UV light nag-cause siya ng dimers. So, yung disease na example natin dito ay yung xeroderma pigmentosum, and we will take a look at this disease later on. So, first, mismatch repair. Ito yung simplified version ng mismatch repair. So, yung mismatch repair, meron kang mismatch ng nucleotide dito, and the mismatch is detected in a newly synthesized DNA daw. And the new DNA strand is cut and the mispaired nucleotide and its neighbors are removed. So, tinanggal. And then, yung missing patch naman na-replace ng bagong nucleotide bases which are correct. And yung replacement ay ginawa ng DNA polymerase mo. And you seal the gap with your DNA ligase. So, yun yung simplified version ng mismatch repair. But actually, if you take a look at mismatch repair, there are proteins involved in your mismatch repair. So, yung proteins mo na important dito are yung mut-S and mut-L. And mut-S and mut-L, they form a complex which binds to the mismatch repair or the mismatch nucleotides. So, mut-S and mut-L, they look out for the mismatch nucleotides. And once they found the incorrect base, magbabind sila doon. And ngayon, na nahanap nila, this protein called mut-H, it looks for a sequence, specifically your GATC sequence, which is near your mismatch. So, kapag nahanap niya na yung GATC sequence, yung mut-H mo, it makes the cut from that GATC sequence. So, yung mut-C mo, magka-cut dito, hanggang dito sa mismatch mo. And once na na-cut na yon, pwede na ulit yung DNA polymerase mo na mag-fill nung vacant regions of your DNA na tinanggal. And then, yung mismatch mo ngayon, mako-correct. Okay? So, after that, yung DNA ligase mo again mag-a-act dito to seal the breaks. So, ayan yung closer or yung proteins involved in your mismatch repair. So, you have again your mute-S-mute-L complex, ito, and then which looks out for the incorrect base, and your mute-H protein which looks for the GAT-C sequence, and from that GAT-C sequence, magkakaroon ka ng excision hanggang doon sa area na merong mismatch. Ganun. So that is for your mismatch repair. So the next one is your base excision repair. So in base excision repair, ito yung simplified version. Or actually, uh, for example, you have here deamination, yung cytosine mo na convert into uracil, right? So in base excision repair, the uracil is detected and removed, and leaving here a baseless nucleotide. And then yung um, sugar phosphate backbone mo din tinatanggal. So now, after mo matanggal yung maling nucleotide dito, pwede ka na ulit maglagay ng bagong nucleotide na correct. So, in base excision repair, dito specifically one nucleotide base pair lang yung tinatanggal, 'di ba? Isa lang. So, yung base excision repair mo, there are actually enzymes involved in the repair. So, yung first mo ay yung DNA glycosylases, which recognizes and removes the unnatural nucleotide. So, flipping out kasi tinatanggal niya yung nucleotide mo na mali. So, tingnan natin ulit dito. So, for example, here, you have a DNA strand. And again, nagkaroon ka ng deamination, which converts your cytosine into uracil. 'Di ba? And then, yung DNA glycosylase mo, yung enzyme mo, siya yung magtatanggal ng uracil. So, dito sa part na to, natanggal na yung uracil mo. And then, yung isa mong enzyme involved is called your AP endonuclease. So, itong AP endonuclease, it removes the sugar phosphate backbone with your phosphodiesterases. So, tinatanggal ng AP endonuclease basically mo yung sugar phosphate backbone mo dito. And now, you will observe na meron kang missing tooth. Kaya, ngayon, um, kapag natanggal na yung nucleotide base pair mo dito na mali, yung DNA polymerase mo can finally add a new nucleotide base na tama. So, ayun, yun yung enzymes involved in your base excision repair. So, yung first mo, yung DNA glycosylase, pwede mo siyang maalala by flipping out kasi siya yung nagtatanggal nung nucleotide mo, yung nucleotide base mo. And yung AP endonuclease mo, siya yung nagtatanggal ng phosphate backbone. So again, 'di ba dito sana dito natanggal ng DNA glycosylase mo yung uracil mo, and dito naman tinanggal ng AP endonuclease mo yung backbone mo. So yun yung base excision repair. Okay. So now let's take a look at nucleotide excision repair. So in nucleotide excision repair daw, yung nire-repair niya ay bulky lesions. So, these bulky lesions ay caused by covalent reactions of the DNA bases with large hydrocarbons such as carcinogen benzopyrene found in tobacco smoke, coal tar, and diesel exhaust. So, bulky lesions mo ay also your pyrimidine dimers caused by sunlight. So, these bulky lesions are bigger or bigger damages in your DNA. And yung repair mechanism involved in removing these bulky lesions is your nucleotide excision repair. So, if we take a look at nucleotide excision repair, ito rin yung basically yung nangyayari. Meron ka ditong bulky lesion in the form of your thymine dimers. So, itong thymine dimers to caused siya by UV radiation. Okay? And then, your nucleotide excision repair, once it detects the bulky lesion, it removes a portion of your DNA strand. And um, kapag natanggal na yon, yung DNA polymerase mo, again, will add the right nucleotide bases. Okay. So, yun yung nucleotide excision repair mo. And these are the enzymes involved in your nucleotide excision repair. So, basically, you have your excision nuclease, which cleaves the portion of the DNA na nagko-contain ng bulky lesion. So here, for example, you have a pyrimidine dimer in the form of cytosine and thymine. Nagkaroon daw ng dimers dito. So ngayon, kailangan to tanggalin, right? Your excision nuclease, it detects the damage and cleaves a portion of the DNA strand na nagko-contain nung dimers mo. And once your excision nuclease detects or removes the or cleaves pala, sorry, cleaves the section of the DNA containing your damage, your DNA helicase, it removes your that section of the DNA. So, itong excision nuclease mo, kinakat lang dito. Kinakat lang dito yung part ng DNA mo na merong damage. Okay? Tapos, yung DNA helicase mo, siya yung nagre-remove actually nung portion na yon. And once na-remove na ng DNA helicase mo yung portion na yon, yung DNA polymerase mo can act again to add the right nucleotide bases. Ayun. So, and lastly, your DNA ligase, it again um, joins the gaps between the strands. So, ito yung enzymes involved in nucleotide excision repair. And remember again that nucleotide excision repair, siya yung main repair mechanism for bulky lesions such as your pyrimidine dimers. So, ayun yung single strand breaks in your DNA. Ngayon, tingnan natin yung double strand breaks. Pero wait lang, inom lang ako. So ngayon, tingnan naman natin yung sa double strand breaks nyo. Okay. Wait. So in repair of double strand breaks, there are two main mechanisms. So the first is your non-homologous end joining, and your next one is your homologous recombination. Wait. So first is your non-homologous end joining. So this is the simplest way to join broken double-stranded DNA together. Why? Because yung dalawang ends lang ng double strand breaks mo, basically, ko-connect mo lang sila. 'Di ba? Or the chromosome is glued back together and usually with a small mutation at the break site. Kaya to tinatawag na quick and dirty. Quick and dirty tong repair na to kasi ginuglo niya lang together yung DNA strands mo. And minsan, nagkakaroon ka ng mutations dun sa site mo. Kasi minsan, yung DNA strands mo doon, sa break na yon, minsan yung ibang nucleotides natatanggal or minsan merong nadadagdag bigla. Tapos pag glue niya back together, nagkakos yun ng mutation in your DNA. So again, the non-homologous end joining is considered as quick and dirty. Kasi nagkakaroon ka dito ng mutation. So, ayun. And ito yung nakita niyong figure kanina, 'di ba? So, basically, experiment 'to ng researchers in Harvard School of Medicine in which they used fluorescence resonance energy transfer or FRET microscopy to observe how non-homologous end joining occurs in your DNA. So, gusto lang nila actually itong ma-observe if kung gaano kabilis and kung paano nagjo-join yung DNA strands mo together. So, as you can see here, nagbabago yung kulay nung um, fluorescent probes, right? From blue to red. So, kapag nagbago yung probes mo from blue to red, ibig sabihin lang nun, nag-connect yung DNA ends mo. So, yung ginawa kasi nila dito, yung dalawang ends ng DNA breaks mo, dito, 'di ba? Meron ka ditong dalawang meron kang double strand breaks and meron ka ngayong dalawang DNA na break. So nilagyan nila ng fluorescent probes dito and dito sa kada end na 'to. And once na nag-bind yung ends na 'to, magbabago yung kulay ng fluorescent probes. So yun yung inobserve nila. Yun yung method na ginawa nila ay fluorescence resonance energy transfer or FRET microscopy. And actually, this uh, opens up further na parang answers to questions on how non-homologous end joining or how DNA repair happens in the cells. Kasi ang alam lang nila dati na in non-homologous end joining, nagjo-join lang yung DNA. Pero dahil ginawa nila 'tong experiment na 'to, na-observe nila kung gaano kabilis or gaano katagal nangyayari ang non-homologous end joining and in which regions of the DNA strand nangyayari siya ng mas mabilis or mas matagal. So, gano'n yung and meron pang other questions sila do'n. So, yun lang basically yung non-homologous end joining and ito yung pinaka-simplest way to repair DNA damage na double strand break. Okay. So, ngayon, tingnan naman natin yung homologous recombination. So, in homologous recombination, meron ka again ditong DNA damage na nagkakaroon ka ng double strand break, 'di ba? Tapos, um, 'di ba meron kang um, alleles in your chromosome and alleles are just the same sections of your DNA with the same genetic information. So, meron ka ditong one allele, pero yung isa mo ditong DNA strand na break. So, you can actually use this strand here or you can use this DNA strand or you can use your DNA here as a template to repair this broken DNA. So, via homologous recombination. So, the damaged region is replaced via recombination using sequences copied from your homolog. Homolog pala yung mas simpleng term. So, itong homolog na to, same lang yung DNA sequences niya dito sa isang homolog. And ginagamit nitong na-damage yung homolog niya to repair itself. So, paano? So, first, you have your double strand breaks, right? And ito yung homolog niya. So, yung nuclease mo ngayon, it digests the 5' ends of the broken strand. So, dinadigest ng nuclease yung region ng DNA na merong double strand breaks. And once it digests your a region of that DNA, magkakaroon ka ng strand exchange by complementary base pairing. So basically, in strand exchange, nag-exchange sila ng DNA strand. So ito 'di ba yung yellow, yan yung may damage and yung portion na 'to na tinanggalan ng complementary DNA nucleotides mo, nag-i-insert siya dito sa homolog niya na maayos. Nag-i-insert siya dun sa homolog niya, sa region ng homolog niya, and then it copies the region of that homolog. So therefore, meron na siya ulit dito na maayos na copy ng DNA mo. And then, once na meron na siyang maayos na copy dito, the invading strand is released and the broken double helix is reformed. So, since meron na siyang maayos na copy dito na kinopya niya doon sa isang DNA strand na homolog niya, pwede na ulit itong ma-repair by copying again the regions of the DNA na maayos. So, dito, pwede yung gap dito, yung ika-copy niya yung nandito kasi maayos naman yun. And since meron ka rin na gap dito, pwede niya namang i-copy yung nandito. And ayun. So the DNA synthesis continues using the strands from the damaged DNA as a template. And again, lastly, your DNA ligase, it connects your DNA strands. So now, through homologous recombination, the double strand break is accurately repaired. So para hindi nyo, ah, meron pa palang isa. So, yung homologous recombination then, it happens in the repair of your broken replication fork. So, as you remember, wait lang. As you can remember, yung replication fork nyo in your DNA replication, ganito yung itsura. But sometimes, your replication fork, nagkakaroon ka rin dito ng damage. And so, through homologous recombination, um, as you can see here, nagkakaroon ka ng first pala, yung nuclease mo, ide-degrade yung part ng replication fork mo na mayroong damage. And then, yung portion mo dito na natanggalan ng complementary nucleotides, magkakaroon ka ng strand exchange doon sa isang strand na maayos pa. And then, the synthesis will continue. So, ayun yung homologous recombination repair sa replication fork mo. So, same lang yung principles niya actually. Yung key step dito ay yung strand exchange. Kasi, ito, para hindi kayo mag-imagine. So, yan yung animation. Wait lang. Ulit. Yun. Damaged DNA. There are proteins involved in your homologous recombination which facilitates your strand exchange. So, meron kang strand exchange dyan na nangyayari. And yung damaged DNA mo, it copies a portion of your undamaged template dito. It copies a portion of your undamaged template. And therefore, magkakaroon ka na maaayos yung damaged DNA mo. So remember again, there are proteins involved in homologous recombination. And di ko na sila iisa-isahin, pero mamaya magbe-mention ako ng protein involved in homologous recombination na kapag may mutation doon, magkakaroon ka ng sakit, specifically cancer. So mamaya makikita nyo. So, sana. Ah, ito na pala. So, now let's take a look at diseases which occurs kapag mayroong dysfunction yung repair mechanisms mo, right? So, remember nyo na when your DNA damages, they accumulate and kapag nag-fail yung lahat ng repair mechanisms mo, magkakaroon ka ng sakit. And ito yung examples ng sakit na nangyayari kapag yung DNA repair mechanism mo nag-fail. So, first is your hereditary non-polyposis colon cancer. So, ito yung Lynch syndrome. Yung Lynch syndrome, siya lang yung general term for hereditary non-polyposis cancer. But your Lynch syndrome, it gives rise to other types of cancer. Pero sa discussion natin, yung specific lang na titingnan natin ay yung hereditary non-polyposis colon cancer. So in hereditary non-polyposis colon cancer, it is a genetic disorder that causes an increased risk of developing cancer. And it is autosomal dominant. And there are mutations in the genes involved in the DNA repair. So, yung specific na mutations dito ay yung sa mismatch repair genes mo or MMR genes. So, di ba, alalahanin nyo yung mismatch repair mo ay isang repair mechanism sa ng DNA mo. And ngayon daw, yung genes mo involved in your mismatch repair, nagkaroon ng mutation. Okay? And once na may mutation, yung mismatch repair genes mo, magkakaroon ka ng microsatellite instability. So, now to diagnose hereditary non-polyposis colon cancer, you can, what you can do is to um, test your microsatellite instability or microsatellite instability testing. So, these microsatellites ay actually part ng DNA mo na repeating sequences siya. And as you can see here, in your normal microsatellites or in the normal replication, yung microsatellites mo normal din. But in your abnormal replication in which meron kang defect in your mismatch repair, kunyari dito, meron kang defect dito, merong variability in the size of your microsatellite. Okay? So, again, kapag yung MMR genes mo, mismatch repair genes mo, ay merong mutation, so, hindi maayos yung pag-repair ng DNA mo, ng mismatch repair mo. Hindi maayos yung function niya. And so, magkakaroon ka ng microsatellite instability, katulad dito. And in microsatellite instability, yung microsatellites mo, they vary in sizes. Okay, so this is actually a way to diagnose this kind of cancer or this kind of cancer called the hereditary non-polyposis colon cancer. So here, as you can see, pwede ka mag-microsatellite instability testing by PCR. So hindi ko pa na-discuss ng in-depth yung PCR kasi sa laboratory ko dapat siya i-discuss. But then, sinabihan ako na lahat muna daw ng lectures niyo tapusin. And sa April, May na lang daw yung laboratory. So, ayun. So, maraming nagtatanong kasi nung na-i-discuss raw yung specific or in detail yung PCR. Pero, ayun. So, i-discuss na lang natin siya kapag laboratory time na natin sa April or May. So, pero basically, ano yung naaalala nyo sa PCR dun sa previous lecture natin? Hmm. Magtatawag ako pero yung hindi naka-open yung camera. Sino kaya? Sino kaya? Hmm. Naka-open ba yung camera mo? Miss, Miss, Miss Rian? Miss Rian. Na siya. Hala, nag-left ng meeting tuloy dahil nagtawag ako. Guys, nahihiya ba kayo mag-recite? Well, sino na lang gusto sabihin yung naaalala nila tungkol sa PCR? Tawagin ko na lang si Ronnie. Ronnie, anong naaalala mo sa PCR last meeting?
Naalala ko po, ma'am, na yung PCR is a very specific and very sensitive test in detecting diseases, especially on the molecular level of testing.
Correct. Yun yung answer na hinahanap ko. So, mabuti na ako si Ronnie. So, yung PCR, sobrang specific niya and siya yung gold standard in your um, molecular biology diagnostics. Pero, bago ako mag-proceed, tawagin niyo yung classmate niyo. Bakit sila nag-left ng meeting? Oh my God. Ha. Okay. So, tawagin niyo ulit yung classmates niyo. Bumalik na sila. Hindi na ako magtatawag ngayon. So, hala, or baka mahina lang yung ano nila, internet or nag-brown out. Well, PCR is a very specific test or siya yung tinatawag na gold standard in molecular biology kasi yung specific DNA sequences niya, yung specific DNA sequences na target mo, pwede niyang ma-amplify. So, yung key na mechanism of your PCR is it amplifies your target sequences and sobrang specific niya. So, in your PCR, actually, there are fluorescent probes. And these fluorescent probes, yung nakikita dito, yung FAM, yung HEX, yung Texas Red. Sila yung example ng fluorescent probes nyo. Pero i-discuss ko pa rin ito in detail sa laboratory nyo. So, ayun. So, yung ngayon, in microsatellite instability testing, meron kang, ang dinedetect mo, yung yung specific sequences in your microsatellites, and yung example dito ay yung BAT6, yung NR24 at yung NR21, NR27, BAT at BAT25. So ito lang naman yung specific um, sequences or parang anong tawag dun? Regions ng sa microsatellites mo. And dito, MSIH or microsatellite instable or microsatellite instability is high. Wait lang, ang dami tuloy umalis. 30. Nag-start tayo ng 40, bakit 30 na lang tayo? Okay, so i-discuss ko na lang tuloy. So, so kapag unstable, kapag high yung instability ng microsatellites mo in your colon cancer, ito yung ma-observe mo. So, in your normal tissue, ganito yung madedetect niya sa real-time PCR. So, sa computer mo, ganito yung ma-amplify. But if you have tumor, meron kang tumor na sa colon cancer mo, ganito yung amplification. So basically, hindi sila nagtutugma, right? Kasi, again, yung microsatellites mo, nagkukos siya ng variability or yung HNPCC mo, yung non-polyposis colon cancer mo, meron kang defect in your mismatch repair which causes instability in your microsatellites. So, okay, okay, nagla-lag lang daw, okay. So, yung microsatellites mo ngayon, meron siyang variable, ano, wait lang, meron siyang variable sizes. So, meron kang microsatellite instability. Wait lang, nagugulo ako. So, ayun. So, again, kapag normal tissue, ganito yung amplification. But yung sa tumor tissue, ganito. So, magkaiba sila. But if you see in your itong MSS, ibig sabihin lang dito, microsatellite stable. So, pagkakaiba niya, stable siya. So, stable dito sa MSS mo. So, nagtutugma yung amplification mo in your normal tissue and in your tumor tissue. So, actually, yung MSS mo and your MSI mo, pwede silang gawing markers. Yun na, naalala ko na yung term dito. Ito, markers to, guys. Markers to ng colon cancer mo. So, ayun. So, if you use if you use PCR to amplify specific regions of your DNA na markers for colon cancer, pwede kang makagawa ng way to diagnose um, hereditary non-polyposis colon cancer. And actually, um, yung cells mo or in certain diseases, specifically cancer, meron talagang microsatellite instability. So, pwede kang maghanap ng markers in breast cancer, for example, or in ovarian cancer, tapos pwede mo yung gawing thesis. So, actually, yung isa kong kaklase sa grad school, yung thesis niya, naghanap siya ng microsatellite in breast cancer. So, naghanap siya ng microsatellites in breast cancer and he used it to diagnose breast cancer. So, yun. So, pwede ka actually maghanap ng microsatellites in different types of cancer. So, maganda yung thesis actually kung papasok kayo sa clinical research in the future. Ayun. So, yun yung sa hereditary non-polyposis colon cancer. Again, wait lang. Meron kang microsatellite instability. Okay? Wait, wait. Mm-hmm. Move on na tayo sa next nating disease called your xeroderma pigmentosum. So, children of the moon kasi hindi sila nakakalabas actually ng umaga kapag wala silang suot na special suit. So, xeroderma pigmentosum, disease siya na nagko-cause ng skin cancer kapag na-expose yung patient or yung tao na merong ganito sa UV radiations or sa sunlight. So, itong mga bata na to, meron silang suot na suit na dinesign ng NASA. So these yung mga suot nila, um, special special garments 'yan or special clothing to protect them from the sunlight. So puwede lang sila lumabas sa araw kapag meron silang suot nito. And these children of the moon, they have what actually happens in them is that the UV radiation causes DNA damage by triggering pyrimidine dimer formations. And this pyrimidine dimer formations, it accumulates in them. Why? Because the excision repair to remove the dimer formation is dysfunctional. So, 'di ba, to remove the to repair these damages, you have to have your excision repair. But in patients with xeroderma pigmentosum, defective yung excision repair nila. And um, due to the mutation in the 7 genes from XPA to XPG, which is responsible for excision repair. So, ayun. Kaya itong mga batang to, hindi sila makakalabas basta-basta kasi nag-accumulate yung DNA damage nila na pyrimidine dimers. So, kapag lumabas sila sa araw, kapag na-expose sila sa araw, yung DNA nila madadamage and yung damage na yun mag-accumulate kasi dysfunctional yung excision repair mechanism. So, hindi mare-repair yung damage na yun. Wait lang. Ayun. So, yun yung cause ng xeroderma pigmentosum. And an alternate form of the xeroderma pigmentosum is caused by the mutation in the 8th gene called the XPV. In this alternate form of xeroderma pigmentosum, it affects the DNA polymerase ETA, responsible for translation synthesis. 'Di ba? Diniscuss natin earlier yung translation synthesis. And ngayon, in uh, patients with xeroderma pigmentosum, na-affect naman yung translation synthesis. So, yung specific na na-affect niya ay yung function ng DNA polymerase ETA mo. So, sa xeroderma pigmentosum patients, meron kang dalawang repair pathways na na-affect. Yung first ay yung excision repair mo and yung isa ay yung translation synthesis mo. So, kaya ngayon nagkakaroon ka ng accumulation of damage in your DNA and therefore um, nagkakaroon ng skin cancer. So, ganito yung itsura ng skin cancer ng patients na may xeroderma pigmentosum. So, di sila makakalabas basta-basta kasi ma-expose lang sila sa araw ng kahit mabilisan lang, magkakaroon na ng damage yung DNA nila and hindi yon mare-repair. So, nagkakaroon sila ng accumulation ng damage. And to diagnose xeroderma pigmentosum, you can do it by skin biopsy or genetic testing. So, kapag, so sobrang rare nitong sakit na to. So, kapag gusto mong malaman kung meron kang or kung meron yung anak mo na xeroderma pigmentosum, pwede ka magpa-genetic testing habang bata pa yung anak mo. So, sa genetic testing, hahanapin yung kung meron kang mutation in your genes responsible for xeroderma pigmentosum. So, dahil rare din tong sakit na to, wala pa masyadong parang gold standard or parang yung pinaka way para ma-diagnose yung xeroderma pigmentosum. But basically, kapag meron kang xeroderma pigmentosum, makikita mo na lang yung onset ng disease and ayun. So, yun yung nangyayari sa patients na may xeroderma pigmentosum. Meron silang accumulation of the defects in your DNA and hindi nare-repair yun and dahil doon nagkakaroon sila ng skin cancer. Okay? Yung next naman ay yung um, genes involved in your homologous recombination repair. yung BRCA1 and BRCA2. 'Di ba kanina nakita niyo, sinabi ko merong proteins involved in your strand exchange in the homologous recombination repair. And isa dun ay yung BRCA1 and BRCA2. So, basically, itong BRCA1 and BRCA2, proteins siya na nag-involve para magkaroon ka ng homologous recombination or magkaroon ka ng strand exchange in your homologous recombination repair. So, if your gene responsible for BRCA1 and BRCA2 is compromised or merong mutations, magkakaroon ka ngayon ng breast and ovarian cancer. So, hereditary siya kasi namamana yung mutation dito sa BRCA1 and BRCA2. So, ngayon, pwede ka actually magpa-test or genetic testing din para makita mo kung meron kang mutation for BRCA1 and BRCA2. Kasi kung meron kang mutation for BRCA1 and BRCA2, high yung chance mo na magkaroon ka ng breast and or ovarian cancer. And again, the BRCA1 and BRCA2, they are responsible for homologous recombination. So, 'di ba, kapag mali or kapag may mutation dito, hindi mag-undergo ng homologous recombination yung DNA strands mo. So, hindi mare-repair yung DNA mo. Ayun. So, to diagnose breast cancer, what you can do is skin needle biopsy or MRI or pwede rin yung um, parang mammogram. Mammogram yung inaalala. Mammogram. So, i-mammogram yung breast mo and makikita kung merong tumor. And also, yung sinabi ko, pwede ka rin magpa-genetic testing and papakita mo kung meron kang mutations in your BRCA1 and BRCA2 gene. Okay. So, ayun lang yung discussion natin. So, to summarize, we discussed about the different types of DNA damage, and we learned the various ways in which DNA repair is conducted for all kinds of damage, such as dimers, single-strand, and double-strand breaks. And we also discussed the mechanism behind DNA recombination repair, and we studied the diseases relating to the dysfunction of DNA repair. So, again, nandito pa rin tayo sa part ng central dogma of molecular biology. And bakit mahaba yung discussion natin sa DNA? Because yung DNA, sobrang important niya kasi siya nga yung may hawak ng hereditary information natin. And we need to study um, all of the things relating to the DNA para malaman natin or ma-relate natin ito in how diseases arise. So we have discussed the DNA structure and DNA replication, and today we discuss DNA repair and recombination. And next time we will discuss transcription and translation. So, ayun lang. Kung meron ba kayong question sa lecture natin? Meron bang question? Hmm. Meron bang magtatanong? Sino yung nagbukas ng ano? Mhm. Sige, isip muna kayo ng tanong. Pero announcement ko muna. 'Di ba sabi ko, nakikita ba ako sa camera? Or masyado ako? 'Di ba sabi ko, last time, mag-meet tayo ng Tuesday. Actually, na-miscalculate ko yung days na kailangan natin. So, actually, 2 lecture na lang pala yung kailangan natin. Pero, yung isa kasing, isang meeting kasi dapat after nito, ilalaan ko sana siya para may basahin kayong um, article tungkol sa cystic fibrosis. But nawalan tayo ng meeting. And yung meeting sana na yun yung Tuesday. So, imbes na mag-meet tayo sa Tuesday at ibibigay ko yung reading na yon, ibibigay ko na lang ngayon yung reading nyo and hindi na tayo magme-meet sa Tuesday. Kasi yung meeting dapat na yon, again, nakalaan dapat siya, nabigyan kayo ng time para basahin yon. Pero nga, nawalan tayo. So, ayun, hindi na lang tayo magme-meet sa Tuesday and ibibigay ko na lang sa inyo yung reading material ngayon or mamaya. 3:30, actually, nakaschedule siyang ma-post ng 3:30. So, ayun. And, ano pa ba? Dalawang meetings na lang para sa lecture yung natitira and dalawa naman na yung meetings natin next week, yung Wednesday and Friday. So, ayun. So, di na tayo mag-meet ng Tuesday. Dahil nga, yung meeting na yun, again, nakalaan siya para dun sa meeting na magbabasa lang kayo. And sakto na, ayun nga, nawalan tayo. So, di na tayo mag-meet ng Tuesday. Bibigay ko na lang yung reading material sa inyo ngayon. Meron ba kayong tanong? Sige. Tignan natin yung PowerPoint. Meron bang tanong dito? Dito? Meron ba kayong tanong dito? Hmm. Kung may tanong kayo, mag-raise lang kayo ng hand or magsabi kayo na mayroong tanong kayo kasi hindi na pala gumagana yung nudge extension. Sad. Okay. Kung wala ng tanong, end na tayo. End na tayo ng session. May tanong ba kayo? Tanungin niyo na ako habang time pa natin kasi kung hindi niyo ako tatanungin, malalabuan pa kayo or something. Diba? Wait lang.
Ma'am, kahit ano pong tanong.
Oo, kahit anong tanong. Basta hindi financial kasi wala rin akong pera, guys. Anong tanong? Ay, sino yung nagsalita tala? Di ko nakita kung anong pangalan. Pinafamiliarize ko pa yung mukha nyo. Pwede po open ang cameras naman po. Opo. Pekenemen. Yung makikilala ko lang si Joanna, si Rian, si Angelica, si Rafael, Mary, si Arian, si Colleen, si Ronnie, mm-hmm. Si Angeline, ngayon. Sila lang yung makikilala ko. Bakit si Christine aso yung ano niya? Yan tuloy yung maalala ko sa'yo. Yung aso mo. Yung itsura mo. Wala na ba kayong tanong? Wait lang, inisip ko yung tanong yung nung kabila. Ano ba yung mga tanong nila? Ah! Itong repair mechanisms na to, may nagtanong kasi kung kailan sila nangyayari. So, itong repair mechanisms na to, nangyayari sila actually ng sabay-sabay in your cells. Kaya mababa yung mutation rates mo, di ba? Kasi sabay-sabay silang nangyayari. So, kahit na biglang magkaroon ng mutation yung DNA mo, yung repair mechanisms na to, sabay-sabay silang nangyayari. And, nangyayari sila bago mapasa nung parent cell mo, yung DNA niya, sa daughter cells mo. So, bago, di ba, alalahanin ninyo yung cell cycle, bago mag-mitosis, nangyayari na yung DNA repair mechanisms mo. So, bago pa mapasa yung DNA mo sa daughter cells mo, nangyayari na tong repair mechanisms mo. So, ayun. So, meron ba kayong tanong? 1 minute. Kung wala na, um, yung kung wala na, may naka-schedule post ako, yung sinabi ko sa inyo, yung reading nyo na dapat nakalaan siya dun sa isang meeting. Mapopost siya mamaya. Cystic fibrosis yun. And dalawa yung naka-attach na file. Yung isang file, yun yung nire-require kong basahin nyo. Yung pangalawang file, PDF file siya, journal article siya na in detail, dinidiscuss niya yung cystic fibrosis, yung molecular mechanisms ng cystic fibrosis. And yung require niya lang required nyo lang basahin ay yung isang file, yung basta nakalagay doon sa post ko kung alin doon. And also, mapo-post ko na yung quiz number 3 nyo. Pwede nyo siyang sagutan again at your own time. Kung kunyari nag-aaral kayo ng 1 AM sa umaga or 3 AM yung trip nyo mag-aral, pwede nyo nang sagutan doon or ayun. O, tapos, wag nyo kalimutan sagutan yung class participation, please. Sagutan niyo yung class participation. Merong class participation form for each lecture. So, sagutan niyo na yung class participation 1 and 2. And mapopost ko na rin yung class participation for lecture 3. And pwede kayo doon magtanong. And kailangan niyo sabihin sa akin kung ano yung natutunan niyo. Kung meron man kayong natutunan sa lecture na to. Para makita ko kung ano yung um, kailangan nating aralin pa or bigyan ng focus. So, kung wala nang tanong, let's end our lecture here. And yun lang. Goodbye. Thank you for listening to my lecture. Um, ayun lang. Bye na, guys.
Bye, ma'am.
Bye, ma'am.
Goodbye,
Bye-bye.
ma'am. Thank you, ma'am.
Bye.
Goodbye.
Bye, ma'am.
Bye-bye. May tanong po ba? Stop recording.